Hi, my name is Jin. We are a very young company. We just started working, and we are closing seed round this month, and hopefully launch our labs in July. So we are developing AI models to design DNA payloads for applications in cell and gene therapy, and we are using our own foundry called ACE Foundry. So we have many problems in current vectors that we use for cell and gene therapy. For example, AAVs are the major workhorse in this field, but has been problematic with immunogenicity, which prevents redosing, but also patient death when a lot of 10 to the 14th genome copy per kilograms are injected to the body. And also problems like patients are dying while they're waiting for manufacturing the cell therapy. So hopefully we are trying to address this problem, coming up with a new type of vectors and designed by AI to explore a larger design space. So the current methods using NGS is that chromosomes are fragmented and cloned in a vector and transfected into the cells, and then you read that barcodes to figure out which DNA sequences are present. But these methods, while it's successful to some degree, has higher potential when you can accommodate the secondary or tertiary structure of chromosomes, because if you fragment it, you lose that kind of information. So using AI models, we are including all those design spaces and using very efficient gene assembly technology called Sidewinder. It's recently published in Nature. We licensed that technology in this field, and using that Sidewinder technology, we are building very clean data sets which can help us to develop new, more efficient AI models. And we are setting up this ACE Foundry in Korea. We are using liquid handlers, but also have a variety of protocols already assembled, and using this kind of workflow to develop our AI models. To give you more information about Sidewinder, it's very efficient because you're not relying on the overlaps that exist within your target sequence. The overlaps are present on the side, so it is sequence independent, which means you can assemble very complex DNA sequences such as high GC content or repetitive sequences. So we incorporating with our biofoundry, we can assemble a large set of DNA vectors in a day, and we can produce our own data sets for training. These are the examples of high GC content. We still get almost 100% accuracy, and also repetitive sequence such as H fibrin, almost over 99%. So we are currently, as a new startup company, we are looking for how we can partner with other companies who has expertise in DNA delivery vehicles, so that we design our DNA and then we can incorporate their delivery vehicles to make a therapy, and also clinical models to drive our in vivo validation, as well as the bioinformatics, where we can explore a vast amount of sequencing data to develop our own AI models. Thank you. Yes.
I had that beta tester for this type of applications, right, in AI, and in the United States. So the question they have is, how do you reduce the hallucination part? Because mathematically, right, the minimum that you have is 15%. How do you take it down?
I think the best way would be to take the data sets that are predicting our goal, but also the sequences that are not producing our goals. So if we take that negative data set, we can also tell them to reduce the whole hallucination.
But if you do that, you have built-in errors in both sides.
I think that's case by case, and I believe that if we can create cleaner data sets, at least we are reducing the manufacturing errors fed into the AI models. We are restricting it to the more mathematical problems, which I do think it is solvable.